this is Dr. Japour, and I'm going to go over the four macromolecules um, that make up everything, all the structures in the cell, and basically those are the building blocks of the cell. And I'm going to start by talking about sugars. Um, However, I think just to make things even clearer, I'm going to add a page. There we go. I just want to remind you the four macromolecules in case this wasn't seen before. So we have four macromolecules. This particular lecture is just on carbohydrates, but we have four macromolecules: the carbohydrates, so we say sugars, uh, you know, that's what it's referring to, carbohydrates, and we're going to talk about what makes a carbohydrate and how they're assembled together and what's their role in the cell. So we're going to talk about that. And the next mini lecture is going to be on lipids. Lipids, we also call them fats. So, and the third lecture is going to be on proteins. So there's no other term for that. Proteins, we all know what proteins are. Well, you might not know exactly what proteins are, but we know they're, found in meat and several types of food where you can, get your protein source. Egg whites, and so proteins. And then finally, nucleic acids. Nucleic acids is the DNA. You might have heard of DNA. DNA is the molecule that houses all of the genetic information that makes up you. DNA and RNA, those are two types of nucleic acids that help with the genetic code and, help the cell make the products that it needs to survive. So we're going to start talking about carbohydrates in this particular lecture. So I just wanted to make sure that you knew there are four macromolecules, and we're looking at carbohydrates in this particular lecture. All right. So in general, organic molecules is referring to, when we say the term organic, we are referring to molecules that contain carbon. Carbon-containing molecules are called organic molecules. There's actually a little bit more to that for the definition. Let me add this here because it's not added on the slide. And hydrogen atoms as well. So this usually goes with carbon. Usually carbon is attached to hydrogen anyway, but I want to make that precise here because, for example, the gas carbon dioxide, which you exhale, is not considered organic, even though it has a carbon in it. CO2, that's a gas. It's carbon dioxide. It has one carbon and two atoms of oxygen. This is a gas that you exhale during cellular respiration, during breathing, right? Inhaling oxygen and exhaling carbon dioxide. It's not considered an organic molecule. So really, technically speaking, in the definition, you need to have hydrogen atoms as well in the molecule for the molecule to be truly organic. Okay, so this is an example. These are examples of organic molecules. You don't need to memorize these, the structures of these molecules. These are just examples. For example, these are examples of organic molecules, not examples of carbohydrates. These are examples of organic molecules. This, for example, is a nucleic acid. This is a nucleic acid. When we get to that chapter, we'll explain, what these pentose sugars are and the nucleotide, each nucleotide, what it is. These are individual monomers that kind of assemble together to create this really large molecule called nucleic acid. But the point here is to show you that it's filled with hydrogens. See all of these H's, and every dot, every corner here, these corners, every corner here is a carbon. Every corner molecule here is a carbon. So this is carbon, hydrogen, and yes, they are also oxygen atoms. And I see a nitrogen over here. So there will be other atoms as well, but because we have carbon and hydrogen, it's an organic molecule. So this is a nucleic acid. These two here are examples of carbohydrates, or sugars. Glucose and mannose happen to be two examples of the many sugars that they are out there. And again, here are the carbons and all of the hydrogens attached to all of these carbons. Yes, we also have oxygen, so that's part of these specific sugars, but the idea here is to show you that those are molecules that contain carbon and hydrogen. Here is an amino acid that, let me see, well, it's two amino acids which form a dipeptide. So it's two amino acids that are strung together at this peptide bond. This is a very strong covalent bond between two amino acids, and it forms a peptide. That's why we call it a peptide bond. So when we get to the chapter of proteins, we will learn that amino acids are the monomers. Amino acids are the monomers of proteins. And sugars here are the monomers of carbohydrates. Okay, and here we have fatty acids. This is a fatty acid, this is another fatty acid, and fatty acids are going to assemble into even larger molecules like a triglyceride, right? Triglyceride. So this is going to be 3 fatty acids on a backbone of glycerol. We'll talk about that in that chapter. But again, so this is going to make up fats. And again, this is to show you that it's made of carbon, and hydrogen. So we have lots of carbon, hydrogen. It's an organic molecule. So all of these molecules that make up the 4 macromolecules are organic molecules. That's what you need to have in your body. That's what makes up the cells in your body. And we start by talking about the carbohydrates. So why carbon? Because it has the capability of bonding with up to 4 atoms, or at least exchanging 4 electrons. And sometimes it will double bond with an atom, but it can form up to 4 covalent bonds with other atoms or functional groups of atoms. And we'll talk about functional groups in a little bit. So it forms this tetrahedral structure when it forms with 4 separate atoms. So here, carbon can bond to 4 other atoms or groups of atoms, making a large variety of molecules possible. And because of that capability of bonding up to 4 different things, it just adds variety. And this is what, the molecules need to be. They need to be varied so we can create so many different larger molecules and create variety in the cell. So everything has a different function, and we can create something very unique with this variety. So here are examples of how carbon is linked to hydrogen. This is methane, CH4. So 1 carbon creating 4 covalent bonds with 4 separate hydrogen. So you would have to go back to the chemistry lecture, the Introduction to Basic Chemistry for Biology lecture that I also have, to kind of review how are these bonds being formed. So this discusses the electrons in the atom and how the ones that are on the valence shell are available for bonding with other atoms. So I'm drawing in black the four electrons in the valence shell of carbon that will create a bond with the 4 electrons that are each individually provided by one hydrogen each. So hydrogen has only one electron in its valence shell. Well, there's only one shell, the first shell, and it's only one electron, and it needs to be up to 2 to be stable. So each hydrogen requires an electron from somebody else. And carbon has its atomic number 8, which means it has 2 electrons on the first shell, and then sorry, I never remember these atomic numbers. It has to be 6 because there's 2 on the first shell, so the remaining 4 are going to end up being on the second shell. And the second shell, for the atom to be stable, it needs to be up to 8 electrons. It needs to fill up, and the maximum it can be is 8. So 4, we are 4 away from 8. So that's why carbon has to, go find 4 additional electrons to fulfill this rule of, having a valence shell that is completed and maximized. So this carbon is going to go find 4 friends that are going to share their electrons with it. And that's what's going to happen between carbon and hydrogen. Hydrogen shares its one lone electron, and carbon will share its 4 electrons on the valence shell. You see these 4 black dots, and those 4 shared with individual hydrogen, and the hydrogen sharing their 1 electron. This satisfies everybody. That way the hydrogens are receiving an electron from the carbon, and that satisfies the here, if I draw this, right, there's only 1 electron on that shell. So one of these 4 electrons from the carbon is going to come, oops, excuse me, is going to come and be shared with the hydrogen. So now the hydrogen will see 2 electrons on its shell, and that satisfies the rule. Okay, so this was a little sidetrack of a review of chemistry, but so each pair of electrons that's shared creates actually a bond, right? And this is how the electrons are shared back and forth like this. And that creates a very strong bond that we call a covalent bond. All right, so carbon can form 4 of these covalent bonds. And one of these bonds doesn't have to be with another hydrogen. It could be with another carbon, and they're sharing 2 electrons with each other. And this other carbon has its own remaining 3 bonds. So this is what we call a functional group. It could be a bunch of other atoms that are stuck on this carbon over here. All right. So carbon is usually bound to hydrogen. There's usually a hydrogen bound. So if it's only bound to hydrogen, it would need 4 of these, 4 hydrogen, because remember, carbon needs 4 electrons. So where is it going to get these 4 electrons? If it's only hydrogen that's available, it needs 4 of hydrogen. If it's oxygen has 2 electrons that are kind of free there. So oxygen has an atomic number 2, and then 4, 6, 8. Yes, 8. So 2 on the first shell, we forget about those, and then 6 on the second shell. 6 is 2 away from 8. And remember, 8 is the maximum, and we need to reach 8 to be stable. So we need 2 more electrons, and this is what's going to happen. We're going to have 2 electrons from shared with another atom that's going to be shared with these 2 electrons. So oxygen can form two bonds. So if carbon can form four bonds and oxygen can form two bonds, we can actually have a molecule like that, and that's actually exactly how carbon dioxide is. It's a carbon in the middle that is bound doubly with one oxygen and double bond with another oxygen, and everybody gets satisfied because you have 1, 2, 3, 4 around the carbon, but 1, 2 electrons shared with this oxygen, 1, 2 electrons shared with this oxygen. Nitrogen has 1, 2, 3 spots available in the valence shell. So nitrogen will need to form 4 bonds. And oops, sorry. 3 bonds. I'm jumping the gun by saying 4 for carbon. And carbon, it's 4 bonds. Okay, it's however many electrons are missing to reach number 8, the maximum number that we can have. That's how many bonds we need to form, because we need to go get these electrons from another atom and form bonds with that other atom. So carbon is used because it can form these large, complex, diverse molecules. These chains, they vary in length. They may be straight, they may be branched, or even arranged in rings like this. Literally. And every corner here, when I talked about corners a couple slides ago, every corner here is a carbon. We just kind of draw it with corners, so we don't. So normally it would be like this, like this, like this. See, it's a carbon, carbon. So that's a covalent bond. So every line here is a covalent bond, and every corner is a carbon. So instead of writing the carbon out, we just kind of like do this. Okay, and of course, these carbons, even though they're attached to two, for example, this one is attached to 2 other carbons, we still need 4 bonds. So there will be here hydrogens on the remaining bonds, because that's, I mean, it could be something else, but I'm just giving you an example here. Carbon has to have 4 bonds. So when you see a corner like that, it is implied that this is a CH2. So each one here is a CH2. If there's nothing else attached, this is a CH2. It's a carbon with 2 hydrogen, carbon with 2 hydrogen, right? So instead of kind of writing it all out like that, we do this. Okay. So back to this image here from earlier. This carbon is, has 1 hydrogen bound to it, but it is forming a bond with another carbon over here, and it's forming a bond with an oxygen over here. So in the end, it does have 1, 2, oh, and another carbon over here. So 1, 2, 3, 4. So that's it. It's satisfied. It's receiving 1 electron from each of these 4 neighbors. And that's what it needs. So molecules can just kind of combine in so many different ways that we end up with this variety. That's really the goal here, is to have a variety of structures, because the different parts of the cell are responsible for different things. So and each job requires a specific setup. This is to show you how these chains of carbon and hydrogen can actually, look different depending on the length of it and what kind of bonds are there. So this is an example of ethane, propane, butane. They're just kind of different names for the different molecules that are of different length. This is a 2-carbon molecule. This is a 3-carbon molecule. This is a 4-carbon molecule. And even though this is a 4-carbon molecule, it's different molecule, 1 butane and 2 butane, because of the arrangement of where the double bond is. This is between the 1st and 2nd carbon. This is between the 2nd and 3rd carbon. So there will be a variety of arrangements, which creates a variety of molecules And that's important because every molecule will have specific properties. So if the double bond is here, this butane 1 will have specific properties that will be different from butane 2 because the bond is somewhere else and it makes the molecule behave differently. Regular butane does not have a double bond in it. It's actually saturated in hydrogen. We use the word saturated, meaning it has all of the maximum of hydrogen possible. Isobutane, cyclohexane, or benzene. Right, so you don't have to memorize all of these different examples. I just wanted to show you how different they can be. So these are called hydrocarbons that are molecules consisting of only carbon and hydrogen atoms. So also why carbon? Because why carbon is so important for organic molecules, and that's why we based the term organic based on that. It's because isomers are possible. So what is the definition of an isomer? Those are molecules that have the same numbers of atoms of the same elements, but different structures and therefore different chemical properties. So how is this possible? Same carbons, same number of atoms, but in the end, the structure is actually a little bit different. So let's look at that. Structural isomers. So it will depend on where the bonds are arranged. Let's look at, well, actually, we just saw examples with butane. Let's look at pentane and 2-methylbutane. Here, you have 1, 2, 3, 4, 5 carbons and 10, 12. So C5H12, that would be the how you write it out. C5H12, that's pentane. Well, that's 5 carbons and 4, 5, 6, 7, 8, 9, 10, 11, 12. My goodness, that's 5 carbons and 12 hydrogens. But it's not the same molecule at all. It's one of the carbons is actually not in line with the others. It's bound to one of the carbons in the middle, and it just literally changes the properties of this molecule. So the number of possible increases as carbon skeleton increases in size. The number of possible arrangements, I should have written here, arrangements. There's a word missing here. Right? The longer the chain is, oh my goodness, there's just then so many different possibilities of putting this carbon here versus here. It's just more possibilities. So more variations the longer these chains are. The second type of isomer, besides being literally structurally different, is called cis or trans isomers. What does that mean? The atoms have the same positions of covalent double bonds. So nothing changes as far as where the bonds are. Same exact bonding pattern. So there's no difference and all of a sudden we have a covalent bond somewhere else. Same number of atoms and the same position of all of these bonds. So what is different? It's the spatial arrangement of the atoms around the double bonds. So usually cis and trans isomers, they exist whenever there's a double bond. When there's a double bond, it kinks the molecule in such a way that whatever's attached on those 2 carbons can actually face upwards and together, or one faces upward and one faces down spatially. So in space, it's not a flat molecule. These groups are kind of like twisted and face either one plane above together or they face opposite planes. So a cis isomer has its 2 kind of the groups that we're comparing. So here we just use the word X, or the letter X. The two X's are on the same side. We use the term a cis isomer, but if these two groups are actually facing, the opposite sides of, the the main molecule chain, then we talk about trans isomers. Okay. And so if you get into biochemistry, you get into in depth about that, and then you'll see how glucose has isomers, just a molecule of glucose. You'll see examples and get more into that. So what are enantiomers? What they are, those are molecules that are mirror images that differ in shape due to the presence of asymmetric carbons. So what is an asymmetric carbon? It's a carbon that's attached to 4 different atoms or groups of atoms, meaning instead of 1 carbon and 4 hydrogens and everybody is like the same, it's the same neighbors on all sides. So we end up with really kind of a very predictable three-dimensional positioning of everything. But when you have completely different neighbors, it's going to be a very specific 3D spatial arrangement. So, for example, we have you see this group and this group, and these 3 carbons in the center, these 3 carbons in the center, but now the groups are in different positions, and we end up with this asymmetry. So again, these are just 2 separate examples. These are not enantiomers between each other. These are just 2 examples of carbons that are attached to 4 different things. So let me go back to that. I don't think that was clear. These are just 2 separate examples of, hey, look at these carbons, and they have 4 different groups attached to them. So let's just look at, for example, this one. This carbon here, okay? This carbon here is attached to this group, CHO, and it's attached to this hydroxyl group that's called hydroxyl and an OH, and it's a link to this entire a group, and then it has this little hydrogen over here. So 4 completely different groups of atoms attached to this one carbon. So 3-dimensionally, it's going to have kind of 2 possibilities that are actually mirror of each other. So this is where the mirroring explanation comes into play here. We call them isomers. Remember, this is kind of going into isomers here. When you are, so if we had a mirror over here, these are mirror images of each other. So if you had your 2 hands, those 2 hands, they are a mirror image of each other, but if you look at them straight on and you put them over each other, they're not the same, right? Your right hand and your left hand are not the same hands. They're oriented in opposite directions, and that's as a result of being mirror of each other. They're a mirror of each other. Usually in biology, the molecules are described as being L isomer or D isomer, and usually only one of the 2 forms is the biologically active form of the molecule. So as if your two hands, really only one works and the other one is just there as its mirror image, but doesn't work. And only one of the hands work. So usually only one in enantiomer is biologically active. For example, in amino acids, it's the L version of the amino acids that's active. In sugars, it's usually the D isomers that are active. So it will depend also on, the type of molecule. So functional groups are groups of atoms that attach to carbon skeleton, and they participate in the reactions or give a unique property to the molecule. And that's basically what helps make this variety of different molecules. And this is a list of functional groups. So for a biology course, you have to know those groups, because this really comes up, throughout biology. When we talk about amino acids in the chapter of proteins, we're going to talk about all the functional groups in the amino acids, and these functional groups give these amino acids different properties that are different from each other, and this helps make every protein unique to the and different from the other. So learn these functional groups. So when you have an OH, that's called a hydroxyl group. And I want to show you the hydroxyl group that was here. You see that? This is a hydroxyl group that's attached to this carbon. So we don't just say, oh, it's just an atom of oxygen attached to carbon. We look at what's beyond it. And if we recognize that this is a functional group, OH is actually together makes up a functional group called hydroxyl, and it gives very specific property to the molecule. So what's that called? That functional group is called an alcohol. So in molecules that have a lot of OH has a lot of alcohol in it. That's what it is. So here's an example of a very small one, ethanol. That's very small, structurally. It's only 2 carbons, and the carbons are attached with hydrogens, except for one of them. One of them is attached to a hydroxyl group, and it makes it. So this makes it an alcohol. With this alcohol group attached to it, ethanol is an alcohol. And ethanol is what's in, hand sanitizers and ethanol wipes. You can kind of disinfect a table with ethanol. We use ethanol to disinfect things. All right, a carbon with a double bond O is called a carbonyl group. And the compound name is called a ketone or an aldehyde. So when we say compound name, we're talking about the molecule. In the end, the molecule will be called an alcohol. So when I say an alcohol group, I really should stick with the actual proper term, hydroxyl. This is a hydroxyl group, and the molecule will be called an alcohol. All right. This is a carbonyl group, and the molecule will be called a ketone or an aldehyde. And you see here's propanol. This is an aldehyde. And because it has this CO right here. We actually have the molecule called propanol. So that would have been a good one to add here as an example of an alcohol versus an aldehyde with a 3-carbon molecule. So this is an ethanol, and usually alcohol have OL at the end of their name. Okay. Carboxyl group have this COOH. So even though there's an OH here, because there's a CO next to it right here, we don't say this is a hydroxyl group. And we don't say that CO is a carbonyl group. If we have an OH here, all of these together are called carboxyl group. And this is what organic acids are made of. So when you see a COOH, it's it's an acid. And this hydrogen over here, you see that hydrogen over here? That hydrogen will have a tendency to kind of like get plucked off and be released in the solution, whatever the solution is inside the cell, whatever the solution is, wherever the aqueous environment is. And it will, the more hydrogen ions, the lower the pH is going to be. And it's going to be, low pH is acidic pH. So that's also something that was discussed in the chemistry lecture. And that's why we call them acids, because it acidifies the solution they're in. When you put an acid inside of an aqueous environment, it will have a tendency to lose that hydrogen, and the more hydrogen ions are dissolved in, well, dissolved, they're released in the solution, the more acidic that solution is. An amino group is made of a nitrogen bound to 2 hydrogen. So here we go. This is an amino group. And the molecule will be called an amine. So glycine is an amine. Sulfhydryl groups are with SH. This is another molecule, an example that has an SH in it. And by the way, glycine and cysteine, they happen to be 2 of our 20 different amino acids. Phosphate group, we're going to talk about phosphate group when we talk about phospholipids in the cell membrane in the next chapter. So phosphate group, very, very important. Glycerol phosphate is going to be a great example. This is going to be the backbone of phospholipids. So in the next chapter, when I talk about phospholipids, you'll have had this introduction to the glycerol phosphate that makes up the phospholipid. Glycerol phosphate is a molecule with a phosphate group at the top. Right here, it's kind of laying down over here. I'm talking about it kind of like vertical phosphate group at the top. And we're going to have 2 fatty acids attached to it on these, on the carbons, on these 2 carbons over here. We'll have fatty acids come attached here, but I will discuss this in the lipid chapter. The methyl group is a very small CH3, and CH3 attaching to whatever it is called, the group is called the methyl group, and the compound will be called a methylated compound. For example, cytosine is methylated on carbon 2 here. Oh, sorry, carbon 5. 1, 2, 3, 4, 5. So it's on carbon 4. And we say 5-methyl cytosine because it's that 5th carbon that's part of the methyl group. All right. All right, so here are examples of 2 hormones, estradiol and testosterone. And look at these 2 hormones. Look at that. Will show you how functional groups really have an impact on the molecule overall. So these molecules are made of carbon, hydrogen, carbon, hydrogen. Remember, every corner here, this is a carbon with hydrogens, because when you don't see any group attached to these carbons, if it's just a point by itself, right? These points by themselves, you assume the leftover bonds that need to be made are with hydrogen. So, for example, here we have 1 bond with the neighboring carbon, 1 bond with this hydroxyl group, and 1 bond with the neighboring carbon over here. So this carbon actually has 1 hydrogen here, because remember, it needs 4 bonds. Okay, so this carbon is attached to a hydroxyl group. And there happens to be another hydroxyl group over here. Look at that. And we have a methyl group over here. Amazing, right? See, we saw these groups. Here, methyl group, that was the last one. And look what we did over here. We left the methyl group over here. We didn't really bother that at all. We still have the hydroxyl group over here and the other and we don't have the other hydroxyl group. So now what we have is a carbonyl group right here, and we have a second methyl group on this carbon over here. We had one methyl group on this carbon over here, and now we have a second methyl group on this carbon over here. So these two additional, well, this is an additional functional group, and this is a different functional group on this carbon over here. These two differences make up two completely different hormones with completely different properties. Estradiol is a female hormone, like it's the equivalent of estrogen. Testosterone, even though females do produce some testosterone, testosterone is a male hormone and with its own properties. Like if you have too much testosterone, you're going to have hair growth, and it's basically what makes up baby boys, males. Testosterone levels rise during puberty, and this is how we end up with building muscle mass and changing the cartilage in the surrounding the trachea, and we end up with Adam's apple in your throat because that cartilage gets more pointy in boys that turn, men during puberty. So completely different properties between these two hormones, and we really only have two differences. That's it. And it's in the functional groups. You see how functional groups can change the function? And look at this. So this is morphine. Okay, this is morphine, and morphine has, so this is the diagram of morphine. This is more like a 3D rendition of that diagram. And yeah, so you can recognize it has carbons at every point here, and we see there's oxygens within the molecule, and a nitrogen over here, a methyl group over here, hydroxyl groups, two hydroxyl groups over here and here. Okay, so it has kind of like this kind of specific setup, right? Look at the difference here. If instead of the hydroxyl group, you end up changing these hydroxyl group into acetyl group. Let me see if okay, this acetyl group wasn't mentioned. This is basically to keep things a bit, because we can add, so many, there are more groups, but these are like the basic ones to remember. But this is now another one here showing you, and you don't have to memorize this one. This is just an example to show, another example to show you, just like I showed you with estrogen and testosterone. This is another example. If you change these two hydroxyl group in morphine, you change these two hydroxyl group and you add acetyl groups, you end up with diacetyl morphine. So this is still morphine, but now it has two acetyl groups, this one and this one. And the properties are completely different. You end up with heroin. That's it. That's the difference between heroin and morphine. It's two diacetyl group that are added on this carbon. So actually, this is how they're added. This, let me show you. This hydrogen is going to be taken off. And instead of a bond between oxygen and hydrogen over here, there's your oxygen. Now it's binding to acetyl groups. So this double bond OCH3 is an acetyl group, and you don't have to memorize that one. Just those in the box above. So this bond over here, but instead of with a hydrogen, now it's with this entire group, and that's it. It changes the entire property of the molecule. You have heroin. And so this was actually invented in a lab. Morphine is a substance that's naturally found in poppy seeds, right? The poppy flower, the red flower, has seeds. If you crush them and you extract the active ingredients in the seed, the active component of it is morphine. Morphine is a natural substance found in poppy seeds. And there's a chemist, a German chemist, that was working in his lab to basically change morphine into an analgesic that we can give patients without the side effect of morphine. Because at the time, early 1900s, do I have a no? Oh no, this is the last slide. It was discovered that morphine was a little bit addictive, and we just needed something different. And heroin was discovered as being more potent than morphine. So heroin was named Hero because it was so much more potent. Potent means stronger. So it was a better analgesic. Analgesic means it takes the pain away. It was a better analgesic. And at the time we were like, this is amazing. Oops, I lost my picture here. It was amazing, a new pain med at the time, and we called it Hero because it was going to be, because it's so much stronger. But at the time, there was no clue about how much more addictive it was. And it was sold in pharmacies on the shelves. And I wish I had a picture. I normally include this picture in some of my presentations about my research, because I work on opioid use disorder, so addiction, and the effects of opioids on the human brain. That's my research. So normally in my presentations, I show a little bit of history and how heroin was discovered, and I show these little vials of heroin that were just sold at the local pharmacy, to take the pain away from even coughing. It was sold as a coughing medicine at the time. Okay, eventually we realized it was so much worse, and it was, regulated and not sold on the shelves. So this is the lecture here is done for the discussion on carbons and introduction to sugars. I'll meet you in the next lecture on proteins.